Hello everyone, my name is Anna Babayan. I'm Cancer Research Marketing Manager at Illumina. 2 weeks ago, I had the pleasure attending and speaking at EACR Liquid Biopsies conference in Lyon. And if you missed that great conference, I would like to give you a chance to listen to my talk in this virtual format. Today, I would like to talk to you about how we as a company enable multiomic approach in liquid biopsy. Illumina is widely known as a manufacturer and provider of NGS instrumentation, but that's not it. We have a huge portfolio of solutions applicable in liquid biopsy research, and today I would like to talk about 3 solutions we added this year to enable even more insights. But first, let's talk about what is multiomics and why it matters. Multiomics usually refers to experiments where more than one ohm are analyzed from the same sample. And why does it matter? We know that for complex diseases, and cancer definitely belongs to one of the best examples for multiomics, it's extremely important to look not only on one ohm, because one ohm, information derived from one ohm, does not explain cancer or any complex disease, but it's needed to look at all levels of central dogma of molecular biology, starting from genomics, over transcriptomics and epigenetics as regulatory levels and up to proteomics to make the picture complete and to understand the biological mechanisms behind. So why would you do multiomic analysis in liquid biopsy? Obviously, by doing multiomic analysis, you can measure causes and consequences, and you can explore genotypes and how they translate into phenotypes and function. So by doing so, you can maximize discovery of your experiment and you can get more insights. When we're talking about multiomics analysis, it's important to be clear on terminology. You can be doing genome, transcriptome, proteome, metagenome, and many other omic analysis, but this is just one dimension. Another dimension is analytical dimension, so what you use as analyte in your experiment. And this can be bulk tissue, spatial experiment, you can be doing single cells, and of course, the topic of our conversation today, liquid biopsy. But there is another dimension which is quite often forgotten, and I really want to stress this. There is a time dimension, and we should consider it, especially in liquid biopsy research, because we all know this is the beauty and the power of liquid biopsy that you can do consequent sampling from a patient. You can take samples at diagnosis, at progression, during therapy. You can take sample today or in 2 weeks, right? And as we're talking about biological systems, as we're talking about human beings, as we're talking about tumors, there is evolution. There is evolution from birth to death. There is evolution from benign disease to progressed disease. There is an evolution from today into state into weeks, and this is actually what we try to capture with liquid biopsy. We are trying to capture the real-time snapshot of the disease, and of course it's different at time point 1 and time point 2. So just something to consider when talking about multiomic analysis in biopsy. As I said in the beginning of my talk, today we will be focusing more on proteomics and genomics. So let's first talk about what we have for liquid biopsy in the genomics. When it comes to ctDNA-based liquid biopsy research, earlier this year, we launched Illumina cell-free DNA prep with enrichment, and I would like to spend a few minutes talking about it. We know that ctDNA can be detected in multiple solid tumors. There are tumors shedding more, there are tumors shedding less, but basically almost all tumors are shedding biological material into circulatory system, which can be detected as ctDNA. And we also know that ctDNA levels are highly dependent on the stage of the disease and some other parameters. So where is liquid biopsy ctDNA-based analysis applicable? Basically at all stages of patient journey, and especially when you do research in cancer, you can have liquid biopsy applications to discover more and have more insights into every stage, actually from early detection or even early screening through therapy selection, minimal residual disease monitoring, and monitoring of tumor progression. The point here is to consider that you need a powerful solution which will reflect the needs of your experiment. And what we're actually seeing is the shift towards custom solutions. The reason behind is that with single gene and non-NGS tests, you're really limited to the method in terms of what you can discover, and a single gene alone, a mutation, does not explain cancer as a complex disease, quite obvious, right? So the same is true for small hotspot panels, whereas they might be good applicable in routine clinical practice, though it's also under debate at the moment. For research, it's not really practical to look at just a defined subset of hotspot mutations. There is definitely need for a broader and more complete picture, and this is where custom solution is highly needed, which gives you flexibility to first go broad and see what is happening on the genomic level, what you can discover in ctDNA, and once you know what mutations you have, you can actually zoom in again with your custom panel, custom solution to look into what really matters. To address this need and enable flexibility, we launched earlier this year Illumina cell-free DNA prep with enrichment. Now it's a busy slide, I appreciate it, so please look at what I'm highlighting. It's open content library prep with flexibility for user-defined content. What does it mean? You can have a custom panel of your choice to look at what matters in your experiment. To give you insights in the performance of this kit, please look at these figures. You need as little as 20 nanogram of cfDNA input, and you can reach down to 0.2 variant allele frequency for SNVs, and you can go down to 0.5 VAF for indel detection and gene rearrangements, and of course you can detect gene amplifications. In terms of workflow, it's quite standard approach. You start from DNA extraction, you do library preparation, sequencing, secondary and tertiary analysis. So that's kind of standard approach, but what really matters is that the same workflow is FFPE compatible, because we all know the situation. At a certain point, latest at the submission of your manuscript, you will be asked whether you have compared results you could see in blood using ctDNA analysis with a primary tissue. And primary tissue, if at all available, is quite often available as FFPE material. So we made the same workflow working with FFPE material. As I mentioned in the beginning of this section, this kit works with a custom panel, giving you the power and the flexibility to design your own panel. But also my Australian colleagues pre-designed the custom panel, which might be also relevant to your experiment. It is a 79 gene solid tumor panel with evidence-based content. And as you can see on the right-hand side in the table, it requires 20 nanogram of input material for cfDNA and 40 nanogram for FFPE material. And it is a full gene coverage panel, so not hotspots, but all 79 genes are covered. Let me quickly guide you through the performance of this pre-designed panel. If you look on the right-hand side in the table, with ctDNA input of as little as 20 nanogram, our colleagues could reach 0.1% variant allele frequency detection with sensitivity more than 90%. And for FFPE material with 40 nanogram input, it was 2.5 variant allele frequency with more than 90% sensitivity. Now, on the left-hand side, you can see the workflow steps depicted. And as I said before, the workflow is fully compatible with FFPE, and the only difference, you would need one additional step to perform DNA fragmentation after you extracted it from your FFPE material. As next, I would like to introduce our solution for proteomics research, Illumina protein prep, and currently we are in the early access limited release phase. A new emerging area in liquid biopsy research is proteomic analysis. And while the idea of measuring protein level in blood is new, you will see in a second why I called it an emerging area. So far, we've been talking about analysis of the genome and other omes such as transcriptome and epigenome being foundational to cell function. Analyzing proteome, we are linking causes and consequences of different layers of regulation and interaction. And again, the idea of measuring protein levels in blood isn't new, but with technology evolving and enabling higher throughput and analysis of thousands of proteins at once, researchers can identify patterns associated with health in different stages of a pathologic process. In this way, the concept of biomarker evolves from being a single measurement point to a signature and thus multiplying power of the analysis. So let me introduce Illumina protein prep. I appreciate, again, this is a very busy slide, so please let's focus first on what I'm highlighting on the bottom of the slide. You need 55 microliters of either serum or blood for this analysis. And we all working in the liquid biopsy space know that 55 microliters of serum or plasma is definitely doable. Sometimes we need a few blood tubes from our patients for liquid biopsy tests. What it offers at the moment, as I said, we are in the early access limited release phase, meaning that our panel covers 6,000 unique human proteins. And as we go towards full commercial release in early 2025, this panel will be enlarged to measure up to 9,000 unique human proteins. And what it offers in terms of performance, it is 10-fold dynamic range, what you can measure, this femtomolar sensitivity and high precision assay. So on the top of the slide, you can see the workflow. It starts, as I said, with serum or plasma. It is a fully automated solution, so it requires a Tecan instrument and a high throughput sequencer, and of course, bioinformatic tools. So how does it work? Illumina protein prep delivers highly sensitive protein capture using SOMAmer reagents and Illumina library preparation. The SOMAscan assay quantitatively transforms the protein epitope availability in a biological sample into a specific signal. Relative protein concentrations are then converted into SOMAmer reagent abundances, and SOMAmer reagent abundances are converted to barcoded, sequencing-ready libraries using a hybridization approach. You can access this solution by either bringing it in-house, or you can also access it through our trusted service sites, and we are working with different service providers to build a network of service providers across the globe to enable proteomic solutions with Illumina. The service provider we are currently having in Europe is ProComcure in Austria, and there will be 2 other service providers in Europe operational by end of 2024. So if you are interested, let us know, reach out to us, and we are happy to advise you. In the final section of my talk, I would like to talk about bioinformatics solution for multiomic data analysis, Partek Flow by Illumina. Partek Flow is a powerful tool for all types of multiomic analysis. You can combine DNA data. Actually, I don't know why it's not depicted on the slide, but trust me, you can use DNA data, you can use RNA data, you can have single-cell data incorporated, spatial, ChIP-seq, ATAC-seq, metagenomics, proteomics, microarray, and actually many more different types of data to combine into analysis. A comprehensive set of analysis tools from start to finish, from QC all the way to biological interpretation and everything in between. This is what Partek Flow offers. And the good thing is that you can access it from anywhere. It is cloud-based solution, and of course, after you've done your statistics and data analysis and everything you wanted to have in your analytical pipeline, you have publication-ready figures, and it is really as simple as mouse clicking and dragging and clicking again. So this is really user-friendly and intuitive solution. And as I said, once you've done the analysis, you have publication-ready figures to put in your manuscript or presentation or whenever you need these illustrations. Coming to the last slide in my presentation, I hope I could convince you that multiomic approach in liquid biopsy is the future of research. And of course, we are seeing this being implemented already now, and more omes and more analytes being incorporated into analysis, and of course powered by AI and sophisticated bioinformatic tools. It gives you even more insights and even more discovery power, and we are always on your side to help you and to equip you with best tools possible. And with that, thank you very much for your attention. And if you would like to have a chat about liquid biopsy, feel free to reach out. I'm always happy to have a chat with you. And thank you once again for your attention. Bye bye.